99% similar as chimpanzees, and if I'm not careful, can I evolve into a chimpanzee?
Well, you are correct, we're very similar to chimpanzees, but fortunately, you're not going to evolve into a chimpanzee. Our species diverged 6 million years ago. And furthermore, to understand our differences, we can talk about genomes. Hey class, welcome back. Today we're going to continue our talk about genomes. So, using comparative genomic studies can help us to better understand our evolutionary past. Specific gene sequences show us that bacteria, archaea, and eukaryotes diverged between 2 and 4 billion years ago. Small numbers of gene differences in closely related species tell us how and maybe even why the genomes were sent on different evolutionary paths. So what genetic differences make us humans and make chimpanzees chimpanzees? Well, there's actually a 2.7% difference due to insertions or deletions of large regions in one of the species. Evolving genes that code for transcription factors tell us a lot about our genetic history and differences to other mammals. These regulate gene expressions, which is why it's so important to study them. And we use them to look at our overall genetic readings. Let's look at a specific example. The FOXP2 gene, or forkhead box protein P2, as pointed out in the New York Times article, Speech Gene Shows Its Bossy Nature, tells us that this gene could be the reason we speak and chimpanzees cannot speak. Results from genetic altering on mice show that when the mouse had one disturbed copy of this gene, or 2 disturbed copies, the mouse struggled to fully develop brain-wise, and both mice disturbed couldn't make any noise at all when under induced stress.
Are we as genetically similar to a chimpanzee as we are to a person who comes from somewhere else in the world?
Not quite. The genetic difference between individual humans is only about 0.1% on average. DNA variation is small in humans compared to other animals because we have a relatively short history of only about 200,000 years. However, just like studying genetic differences between us and other species helps us understand evolution, studying the genetic difference within our own species can help us understand the evolution and history of our species. The most common cause of gene variation in humans comes in the form of single nucleotide polymorphias, or SNPs, or SNPs. These are a variation of a single base pair that occurs in at least 1% of the population. Another form of variation is a copy number variance, CNVs, a place where a gene or gene region has a different number of copies besides 2. Studying these variations and comparing the genomes between populations provides insight into how different human populations developed and migrated in early history. A study done of the genomes of 2 Africans from different communities revealed more genetic variation between one another in those 4 genomes than would be seen between a person who comes from Europe and a person who comes from Asia. Another study, published in the Nature International Journal of Science, analyzed genomic data and came up with a proposed map of migration of early human populations, seen here. The genomic evidence analyzed in this study showed that Africans have the most genetic diversity among groups, which supports the idea that the root of the human genetic tree is in Africa.
2, 3. Would you believe me if I said that fruit flies and humans are more similar than you think? Of course not! Well, with more research about genomic information, it is found that only a few differences in gene sequence and gene regulation account for these huge variations in related species. Scientists have referred to fruit flies as little people with wings. This is due to a special 180 nucleotide sequence called homeobox. Each homeobox contains homeotic genes, or HOX genes for short, which regulate the development of anatomical structures in various organisms. These homeoboxes are widely conserved in animals. Another example of these homeotic genes can be seen when comparing fruit flies and mice. When we look at this picture, the colored bands represent homeotic genes that control the formation of anterior and posterior structures of the body. In fruit flies, these genes are on one chromosome, whereas mice have similar sets of genes except they are on 4 chromosomes. It is important to note, however, that not all homeobox containing genes act as homeotic genes, meaning they do not directly control the identity of body parts, but most of them are associated with development. As previously mentioned, many genes related to development are conserved from species to species. This may be confusing because two very different species could have similar genes but look very different. This is due to small changes in regulatory sequences for particular genes cause changes in gene expression patterns that can lead to major changes in the body form as seen when comparing the brine shrimp and grasshopper. Here we can see how changes in the expression of Hox genes have occurred over evolutionary time since insects diverged from a crustacean ancestor.
In this lesson, we have seen how similar we are when compared with other species based on our genomes, and how comparing our genomes with other species sheds light on our evolutionary history.
Also, we learned about how even though we are very similar to one another, those seemingly small differences in our genes can reveal a lot about humans' past.
These similarities represent a common ancestry of life on Earth and how it has evolved over time. We want to thank you for joining us today. See you next week. Thank you.